Hey guys, this is Mrs. Kraus, and this is our second video on the Part 2 notes, which is about meiosis, the type of cell division that creates sex cells. Okay, so we left off on this slide that talks about the first stage of meiosis, which is prophase 1. And we talked about how homologous chromosomes can exchange sections of DNA during prophase 1 in a process called crossing over. So we are going to go on to talking about each of the stages in meiosis 1. In prophase, synapsis and crossing over do take place. I did make a mistake on your notes, and I said that the chromosomes uncoil into chromatin. It should actually be the other way around. The chromatin has to coil up into chromosomes, just like it does during prophase of mitosis. And once they're coiled up, then synapsis and crossing over take place. Okay, and we see that happening here with our two pairs of homologous chromosomes. They are crossing over. Okay, the nuclear envelope, which we see these little white fragments of their nuclear envelope, is going to break down. And also our centrosomes, which look like little stars, move to opposite ends of the dividing cell, and the mitotic spindle fibers start to grow from the centrosomes. Now, during metaphase 1, we see the pairs of homologous chromosomes line up at the center of the dividing cell along the metaphase plate, and the spindle fibers have attached and actually pushed those chromosomes to the center of the dividing cell. Now, you'll see the word kinetochore in this diagram. You can just ignore that. It's really just the part of the centromere, which is the middle of the X, where the spindle fibers attach. Notice that they're lining up in pairs, which is different from how they lined up during metaphase of mitosis. During metaphase of mitosis, if we had 4 chromosomes, they'd line up 1, 2, 3, 4 in a single file line along the metaphase plate. So this is different. During anaphase 1, the spindle fibers are going to pull homologous chromosomes to opposite ends of the dividing cell. Notice that each of the chromosomes is still replicated. It still looks like an X. Okay. During telophase 1, our homologous chromosomes are going to get to opposite ends of the dividing cell. Now, in some species, those chromosomes will uncoil back into chromatin. But in other species, the chromosomes will stay coiled, especially if in that species the cell goes straight into meiosis 2 and doesn't undergo interkinesis, the period of rest. Now, in some species, the nuclear membranes and nucleoli reform, especially if the cells are going to go through interkinesis. And again, the mitotic spindle will probably break down in the species that go through interkinesis. Now, cytokinesis happens at the same time as telophase 1. It's going to divide the cytoplasm and also separate the organelles into the 2 daughter cells. And this is an animal cell because there's no cell wall. So we see the cell membrane pinching in, and that pinch is called the cleavage furrow. Eventually, it will totally separate these 2 cells. We will end up with 2 daughter cells, and we consider them haploid because they have half the chromosomes of the original parent cell. For this picture, we started out with a parent cell that had 4 chromosomes, 4 replicated chromosomes, and we're going to end up with 2 daughter cells that each have 2 replicated chromosomes. In human cells, we'd end up with haploid daughter cells that have 23 replicated chromosomes. But they didn't want to draw 23 chromosomes in each cell. I get it. All right, now, some species go into interkinesis, some species do not. Either way, once they start meiosis 2, it's going to begin with prophase 2. Now, I've only shown one of the daughter cells of meiosis 1, but remember there are 2 of them. So we start out with these 2 haploid daughter cells that have 23 replicated chromosomes. And again, those are the daughter cells of meiosis 1. Now, if our chromosomes uncoiled into chromatin during telophase 1, they will coil back up again during prophase 2. If the nuclear membrane and then the nucleolus reformed during telophase 1, they're going to break down again. And if the mitotic spindle broke down during telophase 1, it gets reformed by these centrosomes. So the spindle fibers will grow from the two centrosomes. All right, now during metaphase 2, notice that the two chromosomes that are remaining in each daughter cell line up single file, just like they would during mitosis. So they're lining up along the metaphase plate one by one rather than in pairs. If they were in pairs, they would be side by side in this cell. During anaphase 2, the spindle fibers are going to rip apart the chromatids that are in each chromosome, and they're going to pull them to opposite ends of the dividing cell. So remember how in mitosis, when the chromatids split, they almost kind of inverted due to the force that's exerted on them by the spindle fibers. So they kind of look like boomerangs now. Remember, once the chromatids split, we don't call them chromatids anymore. We call them daughter chromosomes. And they are unreplicated because they don't have an attached copy. They don't look like X's. During telophase 2, the chromosomes uncoil into chromatin, our nuclear membranes and nucleoli reform, and the mitotic spindle breaks down. And again, remember, this is happening twice, so we actually have 4 daughter cells. Cytokinesis will occur at the same time to divide the cytoplasm and separate the organelles into the daughter cells. Ultimately, we're going to end up with 4 haploid daughter cells. And each of those daughter cells will have, in humans, 23 unreplicated chromosomes. And in these example cells, they each have 1, 2 unreplicated chromosomes. They're kind of showing that they're unwinding into chromatin, which is why they don't look super clear here. But this would be 1 chromosome, and this would be the second chromosome. They look like lines rather than X's because they are unreplicated. Now, as a reminder, crossing over, which happened during prophase 1, is a way that we can get genetic variation or genetic diversity in the sex cells, eggs and sperm, that are created by men and women. So because crossing over exchanges different sections of DNA each time a woman's ovary cells go through meiosis, no 2 eggs created by that woman will be genetically identical. They'll all be different. Same goes for the sperm created by a man. They will all be different from one another. So even if you have the same parents, you were created with 2 genetically identical or genetically different eggs and sperm, so you are different from your siblings. Now, let's go ahead and talk about another way to increase genetic variation in egg and sperm cells, and that's called independent assortment. So this picture right here is showing a cell that's going through metaphase 1. We know that it's metaphase 1 rather than metaphase 2 because the chromosomes are lining up in pairs rather than in a single file line, 1, 2, 3, 4, 5, 6 along that metaphase plate. Now remember, each pair of chromosomes has a maternal chromosome and a paternal chromosome. The maternal one came from this person's mom, the paternal one came from this person's dad. These chromosome pairs could all line up the same way, in this case with the maternal chromosome on the left and the paternal chromosome on the right. However, they don't have to line up the same way. Independent assortment means that each pair of homologous chromosomes, so this top pair, the middle pair, and the bottom pair, they can all arrange themselves differently along the metaphase plate. So they could line up like this, or they could do something like this, where the top pair has the maternal one on the left, paternal one on the right. The bottom pair does the same thing, but the middle one arranges itself with the paternal chromosome on the left and the maternal chromosome on the right. This is just another one of the possible combinations of maternal and paternal chromosomes in the daughter cells. Now, independent assortment will actually result in 2 to the nth power possible combos of maternal and paternal chromosomes in the daughter cells. Now, for the n is supposed to represent the number of pairs of chromosomes. In this case, we have 3 pairs, 1, 2, 3. So that means we have 2 to the 3rd power possible combinations, or 8 possible combinations. In a real human cell, we have 23 pairs of chromosomes. So that would be 2 to the 23rd power possible combinations of maternal and paternal chromosomes in the daughter cells. That's crazy. So that would end up being over 8 million possible combos. Now, we just talked about the maternal and paternal chromosomes, but how does that actually result in different genes in the daughter cells? Remember that homologous chromosomes have the same types of genes in the same places, but they don't have to have the same gene forms. For example, one homologous chromosome, like the maternal chromosome, might have the blue eye gene and the straight hair gene, and the other one might have brown eye gene and curly hair gene. For example, let's say we have a dividing testes cell, and within this testes cell, we have 2 pairs of homologous chromosomes. We have the A chromosomes, where big A is the maternal chromosome and little a is the paternal chromosome. And we have the B chromosome pair, where big B is the maternal chromosome and little b is the paternal chromosome. Within each chromosome, the 1 and 2 are supposed to just represent the 2 chromatids, or identical copies of DNA. Now notice that within the maternal A chromosome, we've got the blue eye gene, and in the paternal A chromosome, we've got the brown eye gene. Within the maternal B chromosome, we've got a straight hair gene, and within the paternal B chromosome, we've got a curly hair gene. Okay, so here we show a possible arrangement of maternal and paternal chromosomes along the metaphase plate. So both pairs in this example have lined up with the maternal one on the left and the paternal one on the right. Okay. And once these pairs of chromosomes divide, we end up with 2 daughter cells, and this is showing those 2 daughter cells that are going through metaphase 2. So now they're going through meiosis 2. The chromosomes line up single file. I've got both maternal chromosomes in this cell, both paternal chromosomes in this one. And then if these cells go through meiosis 2, we end up with the chromatids, or daughter chromosomes, in 2 of our daughter cells having the maternal gene form. So they both have the blue eye gene and the straight hair gene. The other 2 daughter cells are going to have the paternal gene forms. So they've got the brown eye gene and the curly hair gene. So due to this particular arrangement of chromosomes during metaphase 1, we end up with 2 possible chromosome combinations, so big A and big B, which are both maternal in some of the daughter cells, and little a and little b, which are both paternal in some of the daughter cells. That gave our daughter cells, eggs or sperm, 2 possible gene combos, blue eye gene, straight hair gene, and then the brown eye gene, curly hair gene in some of the daughter cells. Now, we don't have to have both chromosome pairs line up with the maternal chromosome on the left, because remember, each pair arranges itself independently of the others. So this is another example where the maternal chromosome of the top pair is still on the left, but in the bottom pair it's on the right. If we send this cell through meiosis 1, and then these are our daughter cells, this is showing them in metaphase 2, where the chromosomes are lining up single file at the center of the cell. Notice that we now have a mix. We have both maternal and paternal chromosomes in both of our daughter cells. That's going to result in the following possible combinations in the final daughter cells of meiosis 2, the sex cells. So in 2 of our daughter cells, we have the maternal big A chromosome with the blue eye gene and the paternal little b chromosome with the curly hair gene. In our other 2 daughter cells, we have the paternal little a chromosome with the brown eye gene and the maternal big B chromosome with the straight hair gene. So we end up with 2 possible chromosome combinations, maternal big A and paternal little b, or maternal little a and paternal big B. that gives us the possible gene combinations of blue eyes and curly hair or brown eyes and straight hair.